Bismillahirrahmanirrahim. March 24, 9700, paper double two, and it's question 4, 5, and 6. Now, in this video, we continue with question number 4. Now, this is a very interesting question, and I'm sure many of you will find it interesting. Table 4.1 shows a sequence of 12 nucleotides. You can see here 12 nucleotides in the template strand of a short length of a DNA molecule. Template strand is the one which is copied by the mRNA. The corresponding primary transcript and the 4 amino acids coded for by the sequence. The table is incomplete. Complete table 4.1 to show the sequence of nucleotides in the primary transcript that would result from transcription of the short length of DNA. Now basically C is going to be G, A is going to be U, because primary transcript is the mRNA. mRNA does not have T in it. Then it will be G, then T will be A, then A will be U, C will be G. So C, G, so you should write it immediately so that everything is clear. Then T, you know, will be A will be U. So G, U, G, A, U, G, A, G, C, A, G, G, sorry, and U, U, G. Now it says table 4.4 shows all the possible template strand DNA triplets that code for the DNA triplets. It shows the template strand DNA triplet. So this is the one which they are showing you. That code for the amino acids labeled AA1, AA2, amino acid 3, amino acid 4. So what is GUG? Now can you see any GUG here? Yes, it's so the DNA triplets for GUG is what? It's CAC. So you have to look at this. So CAC here is val. Then TAC here is what? Is met. Then TCC. What is TCC? Do we have any TCC here? Yes, here is the TCC. So it's ARG, arginine. Then I'm using a different color so that AAC is here. That is leucine. So it said DNA template strand. Now complete 4.3 to identify the 4 amino acids. So amino acid number 1 would be what? Which is amino acid number 1? Would be what? Would be valine or VAL. Then amino acid 2 would be MET or methionine. And amino acid 3 will be ARG. And then this will be LEU. So this wasn't a very difficult part. And there's only one mark for it. That just shows you how easy it was to score in here. Now it says in part 3, one type of gene mutation is caused by the substitution of a DNA nucleotide. Using the information in table 4.2, state and explain the effect on the final protein substructure of a substitution of the nucleotide at position 3 in the table 4.1. Now let's look at position 3 in table 4.1. Table 3 would be what? CAC. So CAC could become what? CAA. At position 3 could become CAG. Could become CAT. But you see all those CAA, CAG, CAT all code for valine. So you see this was a very good thing which they checked. It says no effect on the protein structure because all the 4 triplets beginning with CA code for valine. CAA, CAG, CAT, CAC all code for valine. So why? Because the genetic code is redundant / degenerate. This is what it means. Redundant degenerate means that 3, 4 different combinations can result. Just the third nucleotide changes, CAC, CAA, CAG. So only the third one is changing. You see, CA is remaining the same. This is remaining the same. So first two nucleotides are remaining the same. The third nucleotide changes, and this is meaning the genetic code is redundant. So no effect on protein structure. All four triplets beginning with CA code for valine. Genetic code is redundant / degenerate. Mutation is caused by the deletion of a DNA nucleotide. Using the information on table 4.2, state and explain the effect on the final protein structure of a deletion of the nucleotide at position 3 in table 4.1. Now let's look at that. At position 3 here, if there is a deletion, and what will happen? So that means we will not read it CAC, we will read it CAT. So CAT will result in the same amino acid, valine. Whether it is CAA, CAG, CAT or CAC, it is the same amino acid valine. So first amino acid is unchanged. First amino acid is unchanged. But then it changes the reading frame. This is called the frame shift mutation. It changes the reading frame because then the reading frame becomes, I'll just use another color. I'll try to get a black color. Then it would be read as this, CAT and then ACT. So this is called a frame shift, CCA and AC, whatever goes in front of it. So this is called a frame shift. So first amino acid will unchanged, changing the reading frame, or you can say frame shift mutation. Then deletion alters all the amino acids after the mutation. So this will change the primary structure of the protein. And it may result in a stop codon causing a premature chain termination, which results in a shorter polypeptide. And this will, of course, result as a changes in the tertiary structure or the active site if it's an enzyme. So first amino acid unchanged changes the reading frame, or this is also called the frame shift mutation, alters all the amino acids after the mutation, stop codon. If it results in a stop codon, will lead to a shorter polypeptide. And of course, then the whole thing is tertiary structure changes, so the active site changes. Now it says replication of nuclear DNA occurs just once in every mitotic cell cycle. 6 named events associated with the mitotic cell cycle are listed. The events are not listed in any particular order. Just draw a circle around each event where replication of nuclear DNA takes place. So that's only interphase and S phase. That's where nuclear DNA replicates. Where would other DNA replicate? Any idea? What happens in G1? Mitochondrial copies are made. So there is circular DNA. So circular DNA will also replicate there. Have you ever thought of that? In G1 phase, organelles replicate. Chloroplast have DNA. Mitochondria have DNA. But it said replication of nuclear DNA occurs. That's a very good question. I would give a question like that to check if you remembered that mitochondrial DNA and chloroplast DNA also replicates. Then outline how DNA is replicated inside the nucleus. That's a very direct question. Double helix unwinds, both strand act as templates. Activated nucleotides, DNA polymerase, leading strand synthesized continuously, lagging strand synthesized in fragments called Okazaki fragments. Then ligase connects or forms the phosphodiester bonds, and this is semi-conservative replication. Then the last question, interesting, shows the structure of an ATP molecule. State the name of the part of the ATP molecule, which is A. What is that? That is ribose, not deoxyribose. ATP has a ribose, and you could not have said pentose because it said the name. So it's a ribose. And for this, of course, I'll give you the mark scheme for this. So the points were double helix unbinds, both strand act as templates, activated free nucleotides align with it. DNA polymerase forms the phosphodiester bonds, leading strand synthesized continuously, lagging strand forms the Okazaki fragments. Then ligase connects the Okazaki fragments, and this is called semi-conservative replication. Then coming to question number 5. The pathogen that causes cholera is a prokaryote. Figure 5.1 shows an electron micrograph of the pathogen that causes cholera. Name the type of electron microscope used to produce the image shown in figure 5.1. Well, of course, this is a scanning electron microscope. Scanning because it's just showing the surface things. It's not showing the details of the inside, which is a transmission electron microscope. So scanning is just going to scan the top of it. So scanning electron microscope, and please do not write the light microscope because we do not have these things in the light microscope. Then name the species of prokaryote that causes, that you have to know is Vibrio, Vibrio, Cholereae. Cholera के end में there's an E at the end. The passage contains a description of the main features of prokaryotic cells. There is one factual error in the message, and the factual error in the question was prokaryote unicellular, generally between 1 and 5 micrometer in diameter, do not have organelles surrounded by a double membrane. They do have cell surface membrane, 70S ribosome, and a cellulose cell wall. This was the mistake. The DNA of a prokaryote is circular and found free in the cytoplasm rather than enclosed in a nuclear envelope. The rest of it is correct. It says identify and correct the factual error. So basically what you had to say was prokaryote cell walls are made up of peptidoglycan. The cells are made up of peptidoglycan, and or you could have said made of murin, not made of cellulose. Not made of cellulose. I'm sure everybody should have been able to pick that up. That wasn't such a very tough one, only a 3-mark question. Then coming to the last question, question number 6. Figure 6.1 is simplified diagram representing a section to the human immunodeficiency virus HIV particle that causes HIV/AIDS. The diagram shows the virus particle is about to attach to the cell surface membrane of a T helper cell at a receptor protein. So this is the receptor protein. You can see cell surface membrane of T receptor, CD4 receptor protein, and CCR5 co-receptor protein. And this thing is going to sort of fit into it. It's going to come here and fit into it. A second protein called CCR5 is also necessary for the virus particle to enter and then infect the T helper cell. So identify structure X. Now structure X is the inside, and this was the viral envelope was outside, which is a phospholipid layer at times. They have a viral envelope. So inside of this would be called the capsid, not caspid. If you wrote that, they wouldn't get a mark for that. And I think a large number of students wrote that. Or you could have said the protein coat. So capsid is the name of it. Yeah, even a capsomere was allowed, or a protein coat was also allowed, but caspid was wrong. It's not Casper the friendly ghost. Explain how the ability of the immune system to resist the damaging effects of a pathogen is affected by discussion of the T helper cell. Basically, whenever they ask you this, you've got to know what do T helper cells do, and then you just have to tell me what they won't do. So T helper cells, produce cytokines. So if the cytokines, there'll be fewer cytokines released. Cytokines are chemicals. And when lesser number of cytokines will be released, so what will happen? There'll be lesser number of plasma cells, so lesser number of antibodies produced, lesser macrophages stimulated, lesser T killer cells stimulated, which will divide and then they kill, so less infected cells will be killed, and a lesser number or fewer memory cells will be produced by the primary response. So basically, whatever the T helper cells would be, we would say that response would be lesser. Lesser number of T helper cells activated, so lesser cytokines released, and the cytokines then result in the plasma cells being activated, and the plasma cells produce antibodies, and they also stimulate the macrophages, they also stimulate the killer T cells, which will punch holes into the infected cell. Now, all this will be less now. So less, 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 less. Everything will be less. Lesser number of memory cells will be produced. So fewer cytokines released, fewer plasma cells, so less antibodies produced, fewer macrophages stimulated, and fewer T killer cells stimulated to divide, fewer memory cells produced. Studies have shown that some individuals did not become infected with HIV even though they were repeatedly exposed to the virus. Later discoveries indicated that these individuals had a mutation in the gene for the CCR5 co-receptor protein. Suggest how mutation of the gene for the CCR5 co-receptor protein provided protection against HIV infection. So you see, if this co-receptor protein, this is the co-receptor protein, this one. Now, if this one didn't have this shape and it had some other shape, it was something like this, then of course this cannot bind to it, this cannot bind to it. So virus cannot enter the T helper cell. So CCR5 unable to trigger the endocytosis of the virus particle, because the gene for the CCR5 protein had a mutation, and the normal shape of the CCR protein was something like this, but then this was something like this. So how would it then, the virus would not be able to attach to it and would not be taken up by the cell. So they will not suffer from HIV. So virus cannot enter the T helper cell, or you could have said CCR5 unable to trigger endocytosis of the viral particle. Now coming to the last part of this question, the use of monoclonal antibodies against the CCR5 receptor protein, anti-CCR5, has been shown to be effective in the treatment of HIV infection. Outline how the CCR5 monoclonal antibodies can be synthesized in the laboratory using the hybridoma method. Now, basically, whatever it is, you just have to give me the steps of the hybridoma method. So, a small mammal or a mouse injected with the antigen, or you could have said CCR5. So, a small mammal injected with the CCR5 slash antigen. Immune response occurs over several weeks. Then plasma cells, actually the mammal is killed and the plasma cells are extracted from the spleen. Or you could have said the B lymphocytes or the B cells are extracted from the spleen. Then the plasma cells or the B lymphocytes are fused with the myeloma cell or the cancer cell to form hybridomas. And then you, of course, screen or you select from the for the hybridoma, which produce the desired antibodies. And then these hybridoma cells are separated, and then large scale production of them, and they produce the clones. So basically these were the six steps which you had to give me. Any three of these were fine, and you gave me actually all this about the hybridoma. So small mammal injected with CCR5, or you could have said the word antigen. Immune response occurs, plasma cells extracted from spleen, plasma cells fused with myeloma cells to form hybridoma, screen for hybridomas and desired antibodies, and hybridoma cells separated to produce clones. And this, of course, is a large scale production, and you get the desired antibodies. That's why it's called monoclonal antibodies.